from Microbe TV. This is Office Hours for what with hiccups for Wednesday, August 7th, 2024. I'm Vincent Racaniello, and you're listening to the viral corner of the internet where you can come and ask your questions about viruses and sometimes other things as well. I have a little hiccups session tonight. Don't know why. It comes and goes, so be patient, folks. Maybe breathing and talking. Give me your clues on how to get rid of hiccups. And don't tell me to hold my nose while I drink a glass of water, right? Someone said it's all about resetting your epiglottis. Anyway, I'd like to know where everyone's from. So start putting that in. But in the meantime, let's thank our moderators who are here tonight. Les is here. Steph San Francisco. Tom Steinberg. Who else is here? Anyone? Vanity Nutrition from Long Island. Thanks, everybody, for coming back. It's very ice water. Well, I'm not going to go upstairs and get ice water right now. Okay, so I got water and I got a cup of tea. Look at this, the mug. This is cool. Does anyone know what TV show this is from? Or actually not a TV show. It's a series. Mornings are for coffee and contemplation. Check it out. I think there were 3 or 4 seasons. The first one was the best. Okay. Told you who the moderators are. What do we have online tonight? We're going to find out where you're from. We're going to talk about microbial matters briefly. I'm going to do a mini lecture on respiratory syncytial virus with a focus on vaccine development and why it failed, but with some biology and pathogenesis there as well. And this was requested last time, you may remember. I don't know if the individual who requested it is here or not. Doesn't matter because this is recorded. You can look later. And you know what? It doesn't matter. It was a good idea. So I made a cute little lecture. Now, sit quietly and breathe deeply. Well, I can't. I have to talk right now, right? A few deep breaths. Let's see where everyone is from here tonight. Okay. We talked about our moderators. Lise is from Columbus, where it's 81F and 2127C. Jan is from Tucson, where it's 90F 23C. Jessica is in Toledo, Ohio, 75, starting to cool down. Kip and Laura. Well, Kip's in the Upper East Side of New York City, where it's 67F with humidity that feels like I'm living in the dog's mouth. Thank you for your support of science communication, Kip. How would you know what it's like to live in a dog's mouth? Have you been there? It's like the Harry Potter film where the kid gives Dumbledore one of those little jelly things, and he eats it, and he says earwax. Like, well, how do you know? Did you ever eat earwax? Anyway, thank you, Kip. Yes, mini lecture today on respiratory syncytial virus. Kang is from Chicago, where it's 22C without any clouds to obscure the setting sun. That's very nice. Clear skies are always good. John is in Minneapolis. 71F 22C Pleasant. Had to chop up a tree that luckily missed my house by a few feet last night. So congratulations on Minnesota. Governor becoming the VP running mate of Kamala Harris. Steph SF. Sunny in 63F. 17C with a light breeze in San Francisco. Looks like a light fog is moving in over downtown now. Patricia's going to watch the Olympics at the same time. No problem with that. Tom is in your Eugene, Oregon, southern Willamette Valley, hot, sunny regional smoke from wildfires. Yeah. Rainy is from the eye of the storm in eastern North Carolina, 78F, 98%. Mark is from Edmonton, Canada, where it's 22C. Christopher Lebowski, I want to say the Big Lebowski when I see your name, in Montreal. MK is in eastern Massachusetts, where it is 66F19C. Pete, 16C, cooler evening in London, heat wave has passed. David Cap is from Dayton, Ohio, 79F26C. We have a lot of a lot of Ohioans here. You know, my hiccups have been quiet until I just started streaming. Maybe I'm short on oxygen, right? Because I'm talking so much. Well, unfortunately, this venue, this media requires me to talk, so I can't stop talking to get rid of. Vanity, hold your breath and count to 10. Do you take in? All right, let's try. It was not as long as I could, but I didn't want to leave you guys just staring at me. SM says, hold it as long as you can. Bay says, cela fait longtemps que je n'ai pas vu une émission en direct. It took many years of French. My pronunciation is not so great, but I really enjoy it. Cela fait longtemps que je n'ai pas vu une émission en direct. Well, welcome. It's been a long time since you've seen a live transmission. Welcome back, BE. Where are you from? Lucy is in Sechelt, BC. The Scientific Libertarian is in Iowa. Patrick is in Bremerton, Washington. Hello, Mark Martin. Sorry, there might have been an echo there. Hello, Mark Martin. Tacoma, Washington, 24.9 degrees C. You see, Mark is a scientist. He goes for the decimal point, one place, the tenths. Bernard is in Tacoma Park, Maryland. I know where that is. I've heard of it. Doreen is 64F, I meant. Almost cuddle weather. Claire is in the UK. Betty is from Washington Heights, New York City. The location of course, Columbia University Medical Center, where I'm still a professor. Rarely go unless I'm teaching. Most of my teaching happens in the Morningside campus, which is south. The medical center is at 168th Street, and then the undergraduate campus is at 116th Street. So I bide my time between the two when I'm teaching virology in the spring term. Carol is in Bakersfield, California, 109F. Had an earthquake last night, lasted only a couple of seconds, no damages. Carol, I say every time, is our favorite nephrologist. Now, if there is another nephrologist here, let me know. We could have two favorite nephrologists. been a long time. Yep. Margot is in LaGrange, Illinois. Margot has a question. It's a good one. I'll come back to that after we finish this. Philip is from Wales in the UK. Okay, to stop hiccups, breathe into a paper bag. Don't put it over your head, and don't use a plastic bag. Limit this to 1 minute. Hold your breath for a few seconds, then exhale. Okay, well, I can't do that here. And I have a paper bag, first of all. I have a huge shopping bag, but that won't work. So you have to put up with this. It usually goes away on its own. This is lasting longer than it usually does. Licio is in Belém, Amazon region, Brazil, 35°C and dry, no rain for 30 days. David takes several small sips of water repeatedly. Let's try that. We can do that. Do we have to repeat that, huh? Speak slowly and deliberately, says Pete. I could do that. I could slow down my usual frenetic pace, right? Nebby is in Lake Wales, Florida, where it's currently 90 degrees Fahrenheit. I tell you what it was here in the burbs of New Jersey. 21°C, drizzling, and our humidity is 87%. Of course, it's drizzling, so it's going to be high, right? Let's try the deep breath. Yeah, I felt one through that. Well, Hat says it's the diaphragm, not the epiglottis. Yeah, that's what I figured. Someone said you could massage your diaphragm. I'll sit here massaging my diaphragm on a Virology live stream. Maybe everyone hitting the like button will solve the hiccups. What do we have here so far for likes? We have 49 likes and there are 105 of you. So yeah, that would be good. Hit the like button. Maybe I'll stop hiccuping. Ariozone is in Asheville, North Carolina, where Hurricane Debbie's grazing us. I bet if I had a really good laugh, I would feel they would go away. By the way, speaking of a laugh, I never remember any jokes, right? But I saw one on Instagram the other day. I thought it was funny. A priest and a rabbi. No, I already blew it. Damn it. All right, here's how it goes. A priest and a rabbit walk into a bar. The bartender says to the rabbit, "What are you doing here?" And the rabbit says, "Well, I'm not sure. I think I'm a spell check error." Here all week, try the veal. Abdul Rahman is back to Brooklyn, 19C. Mary Remy is from Dalton, Illinois. Bonsoir. Drink water from the farther rim of the glass. Yeah. It'll spill down my chest. That's a joke, right? That's funny. GSD Bio Jonathan from Encinitas 25. See it nice. With the exception of a few locales, it seems to be cooling off. Michael is from Mexico City, where it's 19C. Joseph is from Ontario. Miliana, well, interesting. It's Ithaca, New York, 70 and cloudy here. Is this a new face? I don't recall Miliana. I went to college in Ithaca. I went to Cornell University from 1970 to 1974. Yes, I've been here a few years. Silent Runner is from Wichita. 80s and showers for the next few days. Been about 100. Diaphragm. 91F in the San Fernando Valley. How many likes do we have? 65 out of 110. Come on, folks. You can do better. Hiccup marathons. I knew someone who had hiccups for like 6 weeks. 6 weeks. Gaming in HD. Professor. You remember my username? Yeah, I do. Did you take my class? Is that why I remember it? Or is it from here? I mean, I've seen it before. Yeah, you're going to be scared if I get really scared. Like, I'll tell you what would happen to make me really scared. If the power goes out and the stream drops, oh my God, I'd be scared. Well, I wouldn't be scared. I would be upset, right? Cheryl is in Santa Rosa, California. Jeff is in Elgin, Illinois. Hiccups make me so frustrated. Yeah, they do, especially when they persist a long time. Yuxing is from Hangzhou, China, 33C. As hot as the furnace. That is hot. So, Yuxing, you did request the RSV mini lecture, and you're going to get it tonight. There you go. Thank you for doing that. I actually had fun putting it together. Vanity says, every time I see Mark Martin, I think of the Microbial World parody to Material Girl. It's very good. Done by a lab. Yeah, that was really good. Mark says, Vanity, I've tried to get students to do that parody for many years. No joy. They don't do it? Well, you have to have the right group, right? Here's an interesting one. Rio says, trying to make yourself purposefully hiccup works wonders, especially if you have some keep asking you to hiccup. Yeah, right. But I'm here alone in my basement. It's okay. I have all of you. I'm not alone, actually. You are my company for this evening. Laura's in San Francisco. Will be in New York later this month. Come visit. Yes, for sure. We had a patient at our hospital with both SARS-CoV-2 and influenza. Yeah, as Daniel was saying, that you can get both. You can have more than one infection, right? You can have as many infections as you please, actually. That's cool. When was the last time you had hiccups? I can't even remember. I hardly have hiccups. And then this week, they have begun. They're intermittent. Fortunately, I don't seem to hiccup in my sleep. So unless I wake up, and then I rip off a few hiccups and go back to sleep. I don't have a cold or anything like that. I just have some. I just ate, so my sinuses tend to drain after I eat. Hiccup laboratory work before our very eyes. They're always teaching us. Yeah, I knew if I asked you, to give suggestions, you would. You have to drink constantly for about 20 seconds. And Mark says, drink constantly, shots. Not on my live stream. I can't drink constantly for 20 seconds. I couldn't. Let's see. I don't like water enough. I just don't. I mean, on a hot day, water is great. but I've been drinking a lot of water lately. I could put on an immune short to give yourself a chance to relax and recover. Less stress seems to help. Yeah, I'll recover. Thank you very much. It's a good idea, but I don't even know actually how to get a video to give you the sound on this. I've never done it. Yeah. Drink water, hold your breath and count to 10, right? Just drank water. Let's try it again. John says, I'm realizing I don't think I've had hiccups since I quit smoking almost 2 years ago. Oh my God. Ay, ay, ay. All right. Who else is here from where? What's brown and sticky? A stick. Ay, ay, ay. Why did the microbiologist cross the road to get to the other slide? That's very good. I like that very much. Very good. Yeah, you should go see Vanity's YouTube channel. She has some funny. There was one of monkeypox is great, really good with nice. What do you call those characters of yourself? I forgot. Chris is from Denver, Colorado. And John says scientific jokes are experimental. Visto tutti, ciao tutti is in Australia, I believe. Mm-hmm. Sam is 15 and cloudy, but I don't know where. All right. So basically, Sam has been listening to you since I was a second-year bachelor. I went on to do a master's in virology and a PhD in immunology. I think you've met a few of my previous colleagues in Denmark. All the best. So you're in Denmark, okay, where it's 15 and cloudy. Oh, you drink water by leaning over the glass. I got it. All right. I wouldn't want to do that on a live stream. I'd make a mess anyway, and I got equipment here. I don't want to screw it up. I could try it later. Truth or Dare is from La Jolla, California. Try swallowing a teaspoon of dry sugar. How about honey? Is there anything I can do with honey? This is multifloral and clover honey from Trader Joe's. You know, this is we talked about with Mark Martin or with Michael Schmidt. Honey should not be fed to infants because there are microbes in it, right? This is a mixture of honey from USA and Argentina. So we're getting USA and Argentina microbes, microbiomes. Yeah, once you stop thinking about it, it'll leave. That's true. That is actually it's actually a something I've noticed. Yeah, so I'll stop thinking about it. Yeah. Can't watch right now, but as it's making dinner time on the West Coast, but I'll be back to gain wonderful virus knowledge. It's all going to be recorded, so please come back anytime. Reaction to spicy food. I did have a little spicy green beans with my dinner tonight. That could be. Yeah. That could very be. Hello, Rima from Iowa. Welcome back. And I have the hiccups, just to warn you. He didn't have Chinese food from downstairs because he said, no, I didn't have Chinese food. Yeah, drinking in that way changes the orientation of the diaphragm. I bet it does. So I'll try it later if these are continuing. But I suspect at some point when I get into the lecture, I'll focus like a razor and it should go away. I guess viruses are willing to share the host with other viri. Well, of course, we have a huge virome in us, right? Thousands of different viruses are in different parts of our bodies, skin, our mouth, our respiratory tract, everything. Your internal organs, your brain, are full of different viruses. So yeah, they coexist for sure. Also, for hiccups, listen for the airplane. I do like to listen for the airplane. You know, whether I'm inside or outside, and you start to hear the droning, and you say, "Focus in on it." Oh, which way is it? Which way is it coming? Which way is it going? And sometimes I can't find it because it's out of sight range, right? But they're pretty loud. By the way, I saw a very disturbing Instagram short video today about a pilot in 1994 who tried to do a stunt at an air show with a B-52. And he was so close to the ground, it stalled and it immediately crashed into the ground. Everybody was killed on board, or 4 pilots. You can find it. It's very sad. Apparently he was a cowboy and they let him do it. Oh my gosh. Spartavus is in the UK. Hello. Welcome back. Do animals hiccup? That's a good question. We could look it up. We could look it up. Do animals hiccup? Do animals hiccup? Many mammals, humans, of course, dogs, cats, rats, rabbits, horses, lions, tigers, bears, elephants, and shrews. Hiccups are a simple reflex that occurs when the diaphragm contracts subtly, forcing air into the lungs quickly enough to close the epiglottis in the throat. Oh, that's very interesting. So Rima, did you ever hear your cat or anyone with a cat? Did you ever hear them hiccup? Okay, this is the big one. Mark Martin, what is your favorite shot? Bring back the college days in the keg stand. Okay, folks, I admit I have been to retreats in grad school. No, as a professor. And I have done the keg stand on your head. They put the spout in your mouth and give you beer. I've also lied down and they mix a margarita in your mouth. Yeah, I did it because this dude just wanted me to do it. Yeah, this is what someone told me. Drink from a cup with my head upside down. I think that's what the drinking from the far rim and twisting yourself does the same thing. Mark likes coconut rum neat. Maybe your sinuses drain after eating. You're allergic to something you ate. Well, that's all food, pretty much. I don't know. Yeah, I mean, it's not just spicy food, which I happen to have tonight, but pretty much all food. I don't know why. I never really had any allergies, but something else must be going on there. Chris just read I'm wandering from where you're from. We'll get to it. We'll finish. I just read the TwivPic Strange Bedfellows, and there was a chapter about a controversial HSV vaccine trial from 2017. I don't know about it. That's a good point. I should look at it. By the way, I bought Zen and the Art of Motorcycle Maintenance. It was recommended by a listener pick. I really like it. It's very good. I'm sure some of you have read it, right? Oh, so Sam is from Aarhus. So I visited University of Aarhus, or Aarhus University. They taught me how to pronounce it, Aarhus, right? So that's where you're from. Fabulous, Denmark. Wonderful. Please give me a hiccup right now. I can't do it on demand. It's a spontaneous thing. He says it over and over again. You must get the honey from Iowa. It's upstairs. Yes. I don't. I don't want to waste it in doing anything to stop my hiccups. This is very nice honey Rima sent me. local honey. It's lovely. It's got a honeycomb in it. And Rio says, I will stop there. I usually don't stop in person, and the person starts to laugh. Mark says, spores will survive almost anything, including honey. Are there botulism spores in honey? Martin, are there botulism spores in honey? I think we talked about it, but I don't remember the answer. Hello, Will. We're from Changsha. So there are two people from Changsha here tonight. How about that? Will and Yuxing. Welcome both. Professor Special K says, I've always wondered why you can't feed honey to infants and how mead, which is made from honey under anaerobic conditions, doesn't cause botulism. Again, Mark Martin, please weigh in. You're here. I know it's not dinner time yet, so please answer those two questions. Hello, Costella from Ohio. Okay, B didn't like my French. Okay, sorry. Was how it was used to be written before they were awarded the 2016 Capital of Culture. So that joke has been made more than I care to know. Mark says, did you hear about the new viral disease of domestic birds? It's called chirpies. It's one of those canarial diseases. The worst part, it's untweetable. Untweetable. That's very good. Mark, did you just make that up or did you hear it? So hearing the train is very interesting because not too far, just a few miles from here where I am, there's a Conrail track where freight trains traverse, right? And in the summer, it's hard to hear because of all the vegetation. But in the winter, when the leaves are all down, it's very clear. And sometimes at 3 or 4 in the morning, a train will pass. I'll be awake, and you hear the horn that they blow for crossings, grade crossings where cars go over, right? Dot, 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 dot. So 2 long, 2 short, and 1 long. It's very cool. And I listen to how sometimes different conductors put a little twist on it. They go, because they can squeeze the horn, and I love it. And then other guys just go, da, 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 da. They don't care. They're probably bored. So I can tell which conductors are having fun and which ones are being bored. I like it. I really like that. Jessica says her doggies have hiccups and they fart and they burp. Yeah, we're animals. We're one of the non-human animals. Apparently, many of them. I wonder if drumming under your ribs helps get rid of them. I'll try it. I could drum for a while. Wayne Googled evidence-based cure for hiccups. The top result, chlorpromazine. Wow. Approved by FDA for intractable hiccups. Well, mine aren't intractable yet. The scientific libertarian says, my cat is in labor. 6 kittens so far. Oh, how cute. Huh. Eric says, Aarhus was our castle and our keep. Well, I said last week, I think, that I visited in Aarhus University, and I had dinner with some PhD students the night before. And they said, one of them says, well, they all said it was a great place to live. And one guy said it's the best place in the world to live. So it's obviously, it's high on very lists of approvals, you know. So B is from Algeria. I didn't know you spoke French. Well, I took 6 years of French, 4 years in high school and 2 in college. So I learned it pretty well. But I also took a lot of Italian. I was brought up in an Italian-speaking household, and I'm very good at it when I use it. I don't use it all the time, so I'm not good at it all the time. But I try to speak in French. It inevitably morphs into Italian. It's because the Italian went in when I was in those first years, which is so important, right? Here's a good joke from Mark. Why do bacteria like nitrates so much? They're cheaper than day rates. I love it. That's really good. Really, really good. Oh, I love my visit to Denmark. I really did. I want to go back one day and do more podcasts. A lot of great scientists there. I remember Aarhus was a fellow who won the Nobel Prize. He's all celebrated there on the building I went in and did my podcast. His maybe it was actually named after him. So further story from this cat giving birth. There was some random cat that started hanging around our house, never left. Now it's apparently my cat, and it turns out she was pregnant. Of course, double whammy, right? And MN says the cat distribution system in action, right? Soren, yes, I did meet Soren, and I did a TWIV with him. It's late there, I know. Thanks for coming, and watch the rest another time. Goodbye. Aarhus is a very, very, very nice house with 2 cats in the yard, and one of them is pregnant. I could make a line to that, right? You could continue it. Okay, Mark Martin said there can be botulism spores in honey. Thank you, Les. And Margot says, my understanding is there can be botulism spores which infants can't tolerate. That might be right also, yeah. And Mark confirms it. Yes, there are not many C-bot spores in honey, but they are there. Infants need a more developed immune system. That's right. They're poorly developed until 6 months or so. A scientific libertarian, to follow up on this cat arc. I saw her catch a bird midair, then eat it. So we had to take her to the vet to make sure she didn't have parasites. Well, a wild cat will often have them, right? Because they haven't been attended to for a while. What did you find out? You made the chirpies joke up. You're very talented with jokes. Very good. Twiv, come for the science, stay for the jokes, and hit the like button. I don't always tell jokes, but I like proper jokes where you say a priest and a rabbit walked into a bar. Contrary, I liked your accent, but I wrote this more than once and you didn't read it. So I was surprised my comments did not appear. I saw your comments, but I didn't know what they meant. But the last one where you put the 2 faces, that said to me, I don't like your accent, but now, so that got my attention. With respect to train horns, from the old song, the switchman knew by the engine's moan that the man at the throttle was Casey Jones. Yep, that's the same idea. You can tell certain engineers apart. Yeah, when conductors are ringing, I love it. Thanks. Yeah, that's a wonderful sound. I take New Jersey Transit. And they are not very good at ringing bells. Okay, Vanity sent me a bunch of ideas for getting rid of hiccups. One of them, biting into a lemon. Someone said take a lemon wedge and cover it with sugar or bitters and bite into it, or a spoonful of sugar. Pulling on the tongue. It's hard to do, but you know, I've seen the dentist do this trick. I got it. During the cure, the hiccup comes. What's another one? Pulling knees up to the chest for a minute in a seated position. Can't do that. Swallowing air. Gosh, I used to be able to swallow air readily as a kid. I can't do it anymore. Whoa. This has actually been fun. Vincent, have you ever been to Brazil? Yes, I've been twice. And I'm hoping to go again before the end of this year. Mark is very accomplished in the art of the dad jokes. Ratio of actually virology to personal stories. Yeah, I'll stop. I like to loosen it up in the beginning, but let me just find out if there's anyone here. Rip the Comp is from Taos, New Mexico. And that's it. All right. One more item. Remember, Mark just published his 50th Matters Microbial, and he has some stickers and a pin and his image with episode 50. If you want them, I'll send you free. Just send it to RSVP. Send an email to rsvp@microbe.tv with your address, your postal address. We'll ship anywhere. It doesn't matter, and we'll send you some of these and maybe some other stuff too. Okay. So that's that. Now let me do a mini lecture on respiratory syncytial virus. Let's see. We have to set this up here. We have to make a little me, and then we're going to do a screen share. And I'm going to go full screen because last time this worked, but I will wait to see on the actual stream if it comes up. But I'm going to talk about respiratory syncytial virus. This is not a lecture from my course. My course doesn't cover viruses individually. My course covers principles. But this is virology, of course. And I actually, could be the beginning of a full lecture if I added more topics to it. But for tonight, it's really sufficient. So I just want to remind you that infection with this virus is an acute infection. We characterize virus infections according to their duration. Not duration, but yes, it's working. Good. Got my little camera up there. Like acute infections are of defined length. And in this graph, we have, which you've seen before, we have virus production on the y-axis and time on the x. And the blue line is production of virus, and the red is period of disease. And you can see that acute infections are defined. They start and they end either with resolution of the infection or death of the host, unfortunately. But then we have persistent infections of different patterns where the virus is with the host for its lifetime. And so this is not respiratory syncytial virus. This is an acute infection that is eventually resolved within a week or two, in fact. And these, just to remind you, acute infections are common public health problems. They cause epidemics that affect millions of people every year, like influenza virus and norovirus and COVID now, SARS-CoV-2. And they're hard because by the time you feel ill, the infection may be over and has spread. It's certainly the case with SARS-CoV-2 that it has spread by the time you feel ill. But many of the other viruses that cause acute infections. It's the same, and so they're difficult to control, typically by vaccines. Now, here's, oops, we got a typo here. We cannot have typos on a Racaniello slide. Here's a history of RSV control strategies, which I'm going to go through bullet by bullet. But this starts in 1956, when the virus was isolated in chimpanzees with upper respiratory tract infection, and then later detected in children with upper tract infection by Robert Channock, an investigator who I met on many occasions, and he worked at the National Institutes of Health in the U.S. So here's the paper. Another, they have a typo in their title, Recovery of Cytopathogenic Agent from Chimpanzees with Coryza. Coryza, or runny nose. And so this was, what was this? I don't even remember where it was published, but wrong one. I don't remember the journal it was published in, but basically a virus was recovered from throat materials of a chimp with coryza during an epizootic of respiratory disease in a colony of these animals. So there were laboratory animals. The new agent produced changes in tissue culture cytopathic effect, which we've talked about, but was not pathogenic for common lab animals. The donor chimps, as well as other chimps involved, developed antibodies in the months following. Susceptible chimps, following intranasal installation of tissue culture material, in other words, virus infected with the agent, developed clinical coryza and subsequently made antibody. A presumptive etiologic association was established between the new agent and respiratory illness in a lab worker, but has not been implicated in the illnesses of small groups of patients with several common types of respiratory disease. However, a number of humans have antibodies in their serum directed against the coryza agent. So they call this the chimpanzee coryza agent, suggesting these individuals have been experienced infection with the new agent or one related to it. So coryza is spelled Coryza, and they put G up here in the paper. And as I said, Bob Channock and Bernard Reutemann together in 1957 in the American Journal of Epidemiology, published Recovery from Infants with Respiratory Illness of a Virus Related to Chimpanzee Coryza Agent, CCA. And there are two papers. This one describes the isolation properties and characterization. So this study was done to recover new cytopathogenic agents from infants with severe lower tract illness. So that would be bronchopneumonia, bronchiolitis, and laryngotracheobronchitis. During the course of these investigations, two similar viruses were recovered, which were demonstrated to be indistinguishable from an agent associated with an outbreak of coryza in chimpanzees, CCA agent studied in the paper I just showed you. And so they found that this is the same virus, actually. So what do these terms mean that they just bandied about? So here's a diagram of the respiratory tract. We have the upper tract here, right, the nasopharynx and the upper trachea. And these are lined from top to bottom with epithelial cells, of course, of different kinds, mucus-producing cells, ciliated cells. There's a mucus layer over all of them. And many viruses, all viruses initiate infection in the upper tract. And various parts of the upper tract are susceptible. And these are the viruses that can infect the upper tract and cause syndromes that we call rhinitis, pharyngitis, and laryngitis. But many also go down into the lower tract, causing tracheitis, bronchitis, bronchiolitis, and bronchopneumonia, depending on whether they're infecting the trachea, the bronchi, the bronchioles, or the alveoli. You get bronchial pneumonia. Now, one thing I want to point out here is that in many people, we all have a mucociliary elevator. So the ciliated cells lining the entire tract, the cilia beat upwards, and there's a layer of mucus on top. So when viruses get down into the tract, they get trapped in the mucus, and the mucus is pushed back up by this so-called elevator, and then we swallow it. It comes up the trachea and goes down the esophagus as we swallow. You get mucus in your throat and you swallow it. So that's a good defense system, but it's typically not as good in older people, which is why they may be more susceptible to lower tract infections. And there are some people with defects in these ciliary motions, and they can be of any age and may partly explain development of severe respiratory infections. Now, this virus was subsequently called respiratory syncytial virus, or RSV. And I'll explain the name in a moment. So this is an envelope virus. It has a negative stranded RNA inside, which is covered with a nucleoprotein and has an RNA polymerase attached to one end. There is a matrix protein underneath the envelope of the virus to give it rigidity. There's a lipid membrane derived from the host cell, and then three kinds of membrane proteins. The glycoprotein, the spike equivalent, if you will, the fusion protein, a second protein, which catalyzes fusion of the viral and cell membranes, and the SH protein, a couple of copies of that. So why is it called syncytial? You remember when we talked about cytopathic effect, we described syncytia formation as one of the manifestations. What is that? So we have giant cells in infected cell cultures with some viruses. This happens to be measles virus. You can see there's single cells in this culture, but there are some giant cells with many nuclei, and those are called syncytia. And they form when a virus-infected cell, shown here in the upper right, in the panel on the right, the upper part, expresses a fusion protein or produces a fusion protein on the membrane, and then it binds to a second cell that has a receptor for the fusion protein. And those two cells will fuse, and they become a cell with two nuclei, and they can fuse with another cell and another, and then you get a giant syncytium. So that's why it's called respiratory syncytial virus. It is now, we know, the most important viral agent of pediatric lower respiratory tract illness worldwide. It infects everyone of all ages, but causes also severe disease besides in children, elderly, and the immunocompromised. There is one serotype with two antigenic groups. It's spread by large respiratory droplets, not so much those smaller droplets. Fomites, contaminated materials, or close contact. For example, contaminated hands. If you touch someone whose hand has mucus containing the virus on it and you touch your eye or your nose, you will inoculate yourself. Short incubation period, initial replication in the nasopharynx, right, the upper tract, but then spreads to the lower tract via aspiration. So this is not something we do all the time, right? When you swallow, the valve to the respiratory tract closes and the swallowed material goes down your esophagus. But sometimes the timing isn't right and we aspirate, and so we cough when we're swallowing or drinking something, right? If you're not paying attention. But in older people and in babies, aspiration is more common. Babies don't have as good a control over the closing off of the tubes, and the elderly and babies and elderly. The elderly gets compromised as well, and that's why the elderly have one reason why elderly have more common lower tract infections. How about the numbers? 80,000 hospitalizations and 150 deaths per year in children less than 5 years of age in the U.S. In low to middle income countries, over 3 million hospitalizations and 100,000 deaths per year. The mortality rate is about 7 per 100,000 live births. Now, what are the risk factors for severe RSV disease? Well, in children, they include being born prematurely, having chronic lung disease, heart disease, but most cases actually are seen in healthy infants. Most of the cases we're talking about here. Okay, so that is the isolation and properties of the virus. So in 1966, the first RSV vaccine trial was carried out with a formalin inactivated vaccine candidate. This candidate did not provide protection against disease. It didn't provide protection against infection, for sure. It didn't provide protection against disease, but in fact led to enhanced disease in infants. So let's take a look at that. Here's the paper in the American Journal of Epidemiology, 1969. And this was a report on, let's get this out of here so you can read that, the clinical trial. Respiratory syncytial virus disease in infants despite prior administration of antigenic inactivated vaccine. So these children got, the vaccine was grown in cells in culture in the lab. And you can see Bob Chanik is here on this paper. It was formalin inactivated and concentrated, and it was 43% of infant vaccinees responded with an antibody response. 91% had a rise in serum complement fixing antibody, which would generally be a broader response. And then they looked at these infants who were immunized when RS virus became prevalent. The rate of infection was no different. So the vaccine did not prevent infection. But 80% of RS vaccinees required hospitalization at the time of RS infection, whereas only 5% of such infections among the control vaccinees resulted in admission to the hospital. So the control in this case was not no vaccine, but a vaccine against another virus, para-influenza virus, which did not cross-react with RSV. And the illnesses the kids got were pneumonia, bronchiolitis with pneumonia, and some of these children died. So they didn't get protected against either infection or illness, and the illness was more severe in the children who got the vaccine. So these findings resulted in a halt in RSV vaccine development until basically the 1990s. It's really, really tragic. People were afraid to touch it because of this trial. So why did formalin inactivated vaccines, RSV vaccines, fail? Well, first of all, the idea of doing a formalin inactivated vaccine was clearly based on the success with inactivated poliovirus vaccine, but it's clearly a different virus, different structures, and different pathogenesis. Now, you could say, well, influenza inactivated vaccine came first, and maybe that was the inspiration, but also influenza is a different virus as well. But anyway, the hypothesis was that formalin disruption broke the protective antigens, the G and the F protein, the two membrane proteins of the particles. So the formalin treatment disrupted these antigens in some way. So we didn't make neutralizing antibodies. We made non-neutralizing antibodies. These antibodies could bind virus particles, but they had a couple of negative effects. First, the bound antibodies led to deposition of antibody virus complexes in the lung, and they fixed complement. In other words, they activated the complement pathway, which results in damage of cells. So these are two of the outcomes, and this is immunopathology, the immune system having a bad outcome. But this paper, which was published in 2009, really pinpointed at least one molecular mechanism for the failure of this vaccine. This is published in Nature Medicine. Lack of antibody affinity maturation due to poor toll-like receptor stimulation leads to enhanced respiratory syncytial virus disease. So that says it all. Basically, they found, they say here, the widely accepted hypothesis attributed vaccine failure to formalin disruption of protective antigens. Here we show that lack of protection was not due to alterations caused by formalin, but instead to low antibody avidity for protective epitopes. Lack of antibody affinity maturation followed pool toll-like receptor stimulation. This study explains why the inactivated vaccine did not protect the children against severe disease. and in fact led to severe disease, hampering vaccine development for 42 years. So basically, the formalin treatment didn't physically trash the antigens, but rather they cause them to poorly stimulate toll-like receptors, which are needed for a strong antibody response. So that's 2009. Meanwhile, people were starting to look at vaccine development as well. They worked on infectious attenuated vaccines. Instead of they said, no more formalin. We're going to get away with it. Let's mimic OPV, oral polio vaccine, and make an infectious attenuated RSV vaccine, which could be injected or sprayed in the nose. These do not cause enhanced disease, but they didn't protect against severe RSV disease. So none of these have been licensed. More recently, there are new candidates, vaccine candidates of this sort that have been produced by engineering the genome, and they are being tested. But as you'll see, these are kind of behind the whole story right now. Okay, so now what happened in the meantime, since we have no vaccines? There's studies on the structural aspects of the glycoproteins. And then in 1991, it was recognized that protection of babies from maternal IgG antibodies. So in other words, if the mother had anti-RSV IgG antibodies, she would pass them to the child and the child would be protected. So they said, let's make polyclonal intravenous immunoglobulins. I'm going to explain that in a minute. That didn't work. Then they made a hyperimmune immunoglobulin preparation. And then finally, monoclonal antibodies came into the story. So someone wants me to explain toll-like receptor, right? So a toll-like receptor is a membrane protein in cells, mammalian cells, which can be on the plasma membrane or within endosomes. And it recognizes foreign entities like viruses and bacteria. And in the case of the toll-like receptor here, it's recognizing the viral glycoproteins. And that turns on the interferon response, which is needed for a strong antibody response. And the problem here is that the treatment of the virus with formalin destroyed the ability of these G and F proteins to be able to stimulate the toll-like receptor on the plasma membrane. So that led to a poor antibody response. So some passive immune strategies developed in the 90s, as I said, based on the observation that infants in the first month of life are protected against severe disease by maternal antibodies. And then when those antibodies decline in the baby, then the incidence of infection rises and leads to a peak of severe RSV disease about 1 to 2 months of age, because it's already declining enough so that it won't protect you against severe disease any longer. And so what are called polyclonal standard intravenous immunoglobulins were developed. They have antibodies from up to 30,000 plasma donors. So they take donors, they pool the plasma, and then they give it. But in clinical trials, they didn't protect infants from severe disease because the antibody titer was too low. And so another product was made called RSV IgIV, or Respigam, which is this hyperimmune product, but not from just everyone, but only from people with the highest antibody titers against RSV. And they saw a reduction in hospitalization, 24% in the control, 10%. So the hospitalization rates were 24% in the control, 10% in the IgIV group. So this is given intravenously. So although it was licensed, it got limited use because there were some side effects, and the babies have to be infused intravenously every month, a process that takes a couple of hours. And so this really limited its use. But it was a proof of concept saying we can make antibodies that protect babies against severe RSV disease. So then enter the monoclonal antibodies. And so the most widely used one was called palivizumab, or Synagis from MedImmune. This is a monoclonal antibody against the F glycoprotein only, which in a clinical trial led to 45% reduction in hospitalization. And so that's licensed. It was used for many, many years, most likely still is, and it's very effective at preventing severe disease. It's actually nebulized into the respiratory tract. The child is put into a nebulizer chamber and inhales an aerosol version of this. At least that was one way of doing it. They made a second generation, which was humanized. In other words, the first one is a mouse antibody, so it has limited life because we make antibodies against it. But if you humanize it, if you change the amino acid sequence enough to make it look like a human monoclonal, it should last longer. That was called motevizumab, but it was eventually withdrawn due to side effects, and it wasn't any better than palivizumab, so the FDA declined to approve it. Now, speaking of MedImmune company that developed this, I did a TWIV at their facility some time ago, and you can check that out at this link. All right, so what about today? What do we have? Well, back in 2008, Jason McClellan, who's been on TWIV a few times, and Barney Graham. McClellan was a postdoc with Graham, I believe. They had this idea that to get a good immune response against F of RS virus, they should stabilize it in the pre-fusion conformation. So when the F protein is on the surface of the virus, it's in called a pre-fusion conformation. And then when it binds the cell receptor, the protein changes to allow fusion to occur, but it doesn't, when it's post-fusion, it doesn't have the right epitopes for inducing neutralizing antibodies. So they figured out by putting two prolines into the F protein of RSV, they could stabilize it in the pre-fusion state, and they saw that induced more neutralizing antibodies. And so these F pre-fusion stabilized F proteins were subject to clinical trials with encouraging results. And you'll see the outcome in a moment. And of course, these findings were the inspiration for the mRNA vaccines of SARS-CoV-2, where two prolines were added to the spike protein of SARS-CoV-2. So it all began with RSV. So now we have adult vaccines. We have Arexvy from GlaxoSmithKline. This is for adults 60 and over. It is in a clinical trial. It was 94% effective in preventing severe disease, 84% effective in preventing any symptomatic disease, really good numbers. And then from Pfizer, we have Abrysvo for adults 60 and over, 86% effective against severe disease with at least 3 symptoms, 67% effective against symptomatic disease with at least 2 symptoms. And we also have an mRNA vaccine, Mresvia from Moderna for adults 60 and over, 83.7% effective against lower respiratory tract disease. So notice, in all cases, effectiveness is defined against disease, not infection. These vaccines, like all vaccines in humans, do not prevent infection. We also have protection for infants. We have a product called Nirsevimab, which is a monoclonal antibody to the pre-fusion F, stabilized with 2 prolines. This is for babies and some young children. 1 dose in a clinical trial reduces the risk of severe RSV disease by 80%, and it lasts at least 5 months, which is the length of the RSV season. So this is a monoclonal antibody that is given to the babies. It's kind of an echoing of palivizumab. But here it's made to pre-fusion F. And then we have a vaccine that I mentioned, which is licensed for adults over 60. Abrysvo is available to pregnant mothers at weeks 32 to 36 of pregnancy. The mother makes antibodies, and those are passed via the placenta to the developing fetus. It's very, very interesting. And here's a little figure from the CDC showing you the season of RSV. These are the colored bars here, and I believe the red indicates the activity of RSV infections. And the vaccine Abrysvo is recommended during week 32 to 36, in any of these months. But after January really is not going to be very effective because we've reached the peak already. And then nirsevimab is recommended for infants during this time here. And since the season is 5 months old, roughly one dose is sufficient to reduce disease by 80%. So now we have amazing, amazing vaccines of all sorts for RSV. This is a very nice Scientific American article that summarizes this in a simple way by Tara Halley. RSV vaccines are finally here after decades of false start. Decades of failed attempts have given way to several successful vaccines and treatments for RSV. And it's really true. You know, we failed many years ago in the 60s, but we didn't give up. We worked on it because this is a significant human pathogen, and we wanted to know what was going on. So we made the vaccines, we experimented, we figured out what was wrong, and now we have a lot of vaccines. And in fact, some other ones are still in development, so it may even get better. So we're going to make a nice dent in this morbidity and mortality caused by RSV. So there you go with your little history of vaccinology, why the vaccine failed, how we figured it out, and how we built upon that to make better products. So let's go back to your questions now, and we can spend these last 40 minutes or so doing that. Let me go back and find some. Yeah, I'm not going to say what has happened here because if I do, it will come back. So thank you for I told you when I started focusing on the lecture, it would be okay. Okay, Pete has picked up a news snippet. UCSF have got to the bottom of the MISC rare events in child COVID. They make killer T cells to a section of N, which matches a cell protein called SNX8. Yes. In fact, we had those authors on TWIV at least 2 years ago where they were talking about this preliminary data. And I just wonder why it's getting in the news now. Maybe they've gone further with it or something like that. But yeah, that's absolutely right. Let's see. Another ones. There's a lot of advice here. Thank you very much. Lots of good jokes. Lots of good remedies. I know there are more questions down. And then we got on our sidetracks here. Okay, here we go. Tony says, I recently had COVID a week ago, but hung out with my friend who didn't seem concerned because his blood type is ORH negative. Is it true O-neg patients don't contract airborne viruses like others? Well, early in the COVID outbreak, there was some indication that blood type conferred some form of resistance, but it was subsequently shown not to be true. So basically, there's no basis for saying that your blood type is going to help you survive COVID. No. What's your opinion of the current HIV vaccine news? They are using BNABs. My question is, haven't we been here before? So, I mean, the BNAB is the broadly neutralizing monoclonal antibody. These are all isolated from infected patients. They're very rare. They show you that such antibodies can exist. And to use it for therapy is very difficult. First of all, you have to mix a few different BNAbs because the single epitopes eventually you'll get resistant to those BNAbs. You have to mix a few, but you also would have to dose repeatedly to prevent infection. And I'm not sure that they are up to that yet, being able to prevent infection sufficiently so you don't establish a latent reservoir of the virus, which takes about 2 weeks. The real issue is, how do we get people to make these antibodies so they're not rare? Can we design a vaccine that will induce the formation of broadly neutralizing monoclonals? That's the holy grail, in my opinion, and that's what a lot of people are working on. But yeah, vaccination, traditional active vaccination is not working because vaccines don't prevent infection, right? And in the case of HIV, you have to prevent infection because otherwise they infect T cells and integrate their genome, and they don't kill the T cell and they last forever. So that's the establishment of the latent reservoir. B wants to know, why are we seeing an increase in tuberculosis cases after corona? I don't know that I think it's a rebound from corona, right? Where there was a lot of isolation and lockdown, and so the numbers went down. I don't see any mechanism, biological mechanism, that is responsible for this. I just think it's like many other pathogens. You saw an increase after COVID because they were down so low during COVID. Tony says we're going to see a lot of extinct, in quotes, infections because of all the U.S. newcomers. Perhaps, but I don't know if that's the right number, but I don't really know this. And it could be that we ensure that they're vaccinated before, say, they're released into the general population. I guess that disturbed me enough so that I'm back to hiccuping enough. Again, Hat says, I've been listening to you for years, reviewing studies, cohort, RCT observational, and the first time you need therapy, you go for hiccups, you go with antidotes. Kind of funny. Yeah. Well, I wanted a quick fix because there's so many. I mean, there's only one FDA approved therapy, right? And I'm not going to get it now, so I have to try an anecdote. It's not an infectious disease, right? So it's not my wheelhouse either. See, chlorpromazine, FDA approved for intractable hiccups. Well, I wouldn't get that anyway because I don't have intractable hiccups. I just have. I think I noticed that when I eat, which I had dinner a half hour before the stream, that kind of triggers them. Otherwise, it's pretty quiet. So maybe my stomach extended and pushed up the diaphragm or down or whatever and screwed it in some way. I don't know. Looking for an explanation. Let's find some more questions. Okay, here's one from Dr. McPharma. I had a crazy idea you might consider measles immunity amnesia. Can we list measles as a select agent? Measles virus. So just to remind you what measles causes amnesia. When you get, a study was done in the Netherlands showing that children who got measles because they were unvaccinated, they lost their immune memory to previous childhood infections and vaccines, which makes them susceptible to infectious diseases until they restore that memory. Now, it's not permanently destroyed because you can revaccinate and protect them. So I think for that reason, it's not a select agent because you can restore the immunity by vaccination. That would be my opinion, yeah. So there you go. Anyone have an opinion on whether end of breastfeeding should affect when infant vaccination starts? Oh, there's more than an opinion. There's some science. And in fact, we did a paper on a recent TWIV trying to figure out for measles, mumps, rubella vaccine, particularly the measles component, when would be the best time after birth to get it? Because the mother gives antibodies to the child. And so you want to wait long enough, up to a year, so that there's no interference of maternal antibodies with the vaccine, because it's an infectious vaccine, right? But as you wait longer and longer, there's an increased risk of infection and getting severe disease. So you have to balance it. And this is a paper that we did. I'll tell you what it was. Let's TWIV measles. Let's try that. Nope, that's too broad. Let's see, measles timing. Nope, I can't find it quickly. But we did a TWIV recently covering a paper that did just that, and they concluded that 12 months would be the best time with minimal risk of getting infected. But that can be done with other viruses using the methodology they develop. It hasn't been, but yes, you could do it scientifically. Yeah. So Sam from Aarhus says it was Jens Christian School, and you were in the school building. I was. Good night. That was the TWIV we did in Aarhus University. Sorry about the hiccups. Good night, Patricia. Yeah, I'll be fine. Hold your tongue and say, I was born in a pirate ship. Is that going to say something terrible? Shit. Yeah, shit. Yeah, you got me to say ship, and they're still there. But it was just a joke. Is my truth or dare? It might be a Daniel question, but many who got first XBB vax 1023 are waiting for Novavax. Initially, rules different for those who had previous mRNA had to wait 1 year. No, you can get Novavax now. If you go to the CDC site, as Daniel has pointed out, you can take Novavax now. Oh, here's a joke from Jessica. Why was the cucumber mad? Because it was in a pickle. Scientific Libertarian had 6 kittens, 2 black and white ones, 1 white one, 2 orange ones, and a skunk. Congratulations. Talia 7 from Hello from the Philippines. John reminds us that RSV should not be confused with Rous sarcoma virus. So there has been an attempt to do that. Typically, our abbreviation for Rous sarcoma virus is RS virus. And for RSV, it's RSV. Respiratory syncytial virus, it's RSV. But people still get them confused, right? Did intradermal inoculation enhance mucosal secretory immunity? No, I don't know what, if you're just talking in general or for a specific vaccine, but no. But, maybe you're thinking of the tuberculin time test, which is done intradermally to determine if you have T cells against tuberculosis bacterium. But all the mucosal surfaces seem to be connected, so you can immunize one mucosal surface and The other makes antibodies as well. Does mother's milk provide RSV protection? Yes. You can also get antibodies to RSV in mother's milk, right? So that's highly recommended, because if you continue to breastfeed past the time that the antibodies that you have given transplacentally are effective, you can provide more months of protection. Although now we can do the vaccination, right? Are immune products derived from ordinary blood donors? Yes. They're specific volunteers who give, a liter of blood or whatever, and then the plasma is made, and many of them are pooled without checking for RSV antibodies. The assumption is made that everyone has them, but it's clearly not right. And that's why they tried titrating the antibodies, picking the highest levels and pooling those, but it didn't work either. Did you hear about the pancreatic cancer mRNA vaccine? I have. Let's look it up. An mRNA vaccine to treat pancreatic cancer. Yeah. A personalized mRNA vaccine against pancreatic cancer created a strong anti-tumor response in half the participants in a small study, and it will be subsequently tested in a larger clinical trial. And in April, there was an article on the Sloan Kettering site. This is Dr. Vinod Balachandran, who has treated patients with mRNA vaccines, and it was developed with BioNTech, right? And the phase 1 was reported in Nature. The vaccine was well tolerated, activated immune cells in half of treated patients. And so we don't know anything about longevity yet. Pancreatic cancer patients, only 12% live 5 years. So that's the number you want to look at. So you have a cohort of patients that have been immunized, and you have to wait, and maybe you report at 1 year, at 2 years, 3 years who's left. Yeah, they just looked at immune responses. And they noticed that the vaccines induced T cells which persisted for 2 to 3 years after treatment. So they're now doing a phase 2 trial and seeing how it compares to the standard of care. So there's a very nice summary on the Sloan Kettering website. Let me see if I can put the link in. It won't be too long. There you go. Thank you for that. That's a good question. Another cricket music night here from Laura. There's a specific temperature. They stop playing their violins every fall till it gets that cold again. They're busy playing every night. Interesting. Thank you. Yu Xing liked the RSV mini lecture. Thank you. I'm glad you liked it. It was a good idea. John donated by the link here. John, so you can go to microbetv.contribute, or you can go to Venmo at microbetv if you want to donate, or you can donate here is a super chat. There's a button below the video screen. Either way, it helps us to continue our work at microbetv. We're a nonprofit entity, and we depend on your donations to do our work. I decided a while ago not to use the advertising route. Excuse me. I thought this would be. I'm struggling with the wire for my earplugs, which is pulling them out of my ear. Thank you. So thank you for your support, John. Really appreciate it. I'm glad you liked the mini lecture. A teaspoon of sugar. I'm going to try that when I'm done. Okay. I wonder if I should put it on a lemon. Vista wants to know if monoclonals are made by copying antibodies people make. So basically, you could say that, but what you do is you take a person who has AIDS, and you assume they have antibodies to the virus, and you show that they do, and then you identify single B cells. So a single B cell makes one antibody against one epitope. And you pull out from these patients' blood single B cells that bind to many, well, say bind to the spike protein, the glycoprotein of HIV. And then you go through all of those antibodies and see which have a broadly neutralizing function. And you do that by taking the genes for the antibodies out of each B cell and then making DNA copies of them and then producing the protein in separate cells in large quantities. So, yeah, you're basically copying the antibodies that people are making. And these broadly neutralizing antibodies are very rare. They take a long time to develop because they require a lot of what we call somatic hypermutation and affinity selection. And Vista says the only rectal digital manipulation. I don't know why that would work. No, I don't think so. John says even though ivermectin had no evidence of being beneficial for COVID, did we see any drop in parasitic diseases in regions that normally experience those parasites? I have not seen a study of that sort, John. If you want to do a proper study, you can't just do an observational study like this, right? You'd have to do a proper clinical study. And they did, but they did it for COVID, not for parasitic disease. It would have been interesting. But then you would have to have a population at risk for infection with parasitic diseases. And, there are plenty in the U.S., but I think nobody has done that study, no. Electron scanning microscopy has helped a lot for understanding viruses. Absolutely. First EM, electron microscopy, then scanning EM have done a lot. And now, of course, cryo-EM, which uses traditional EM with frozen specimens, lets us solve structures in months instead of years. It's amazing. Is August when they begin to choose what flu vaccines will contain for the fall? I actually have a slide on that. And if you give me like 2 minutes, I believe I can find it because that would be the vaccine lecture, which would be pretty late in the course. Let's see. Is it lecture 20? Antivirals, so that means it's 19. Okay, so let's open up lecture 19. And you want to know the timing. There you go. Here it is, John. You ask to jump, and I say how high. So here is selecting an influenza virus vaccine. So we start to vaccinate September, October, November, December. So August is in the packaging segment. So it's already been selected. The strains were selected back in January and February, and it took till August to get them out. So there you go. Straight from the lecture. Thank you for asking that. MK asks, if mom is known to be not immune to measles, how early can the infant be immunized? Well, it could be immunized within months, right? But knowing that the mom is not immune is not easy. You'd have to do an antibody titer, which you could do, but it isn't routinely done, and I don't know if an obstetrician would do it. But I don't think they will change the schedule because the ACIP has an immunization schedule. I'm not sure a pediatrician would be able to do that, but it would be up to them. So in theory, yes, let's just say in theory, without any clinical practice commentary. In theory, you could do it. Yeah, you could do it earlier. Shan is from Tasmania, Australia. And a TWIP, perhaps in 2022, Dixon showed an ectoparasite that he described as the tank, a black sphere with 2 stabilizer feet. Could you possibly find that information for me? An early 1800 building has been open for occupation, and unfortunately some ectoparasites have come from the original rock walls, and especially when rooms have been heated. All right, I'll look for it. Let me write it down. So you think it was in TWIP? I should be able to find that for Shen. TWIP 2022 maybe, and Dixon ectoparasite. Black sphere. You know, Dixon may just be able to say it offhand, so I'll ask him first. Well, I haven't been in touch with him lately. Eric says, question, if I was breastfeeding for over 12 months, can you immunize later? If no breastfeeding, immunize earlier. We didn't cover it, no. I don't believe it was, excuse me, covered in the paper. But they did say breastfeeding would add to the interference, yes, but you know, you have to, once you get past 12 months, then you have a higher risk of getting infected, right? So you have to balance that. I just don't know. I mean, in theory, yes, but you'd have to do the studies to allow you to make these changes, right? Yep. Tim says Charles Osborne had hiccups for 68 years. I'm not going to have hiccups for 68 years. There's a TWIP about the benefits of parasites in preventing COVID. Apparently, parasites don't want their host to die. Actually, I think it's that parasites have been selected that manipulate the host immune response to benefit them, and that may also benefit other infections as well. So I don't think it has to do with them wanting anything at all. Yeah. Would you consider inviting Dixon back for an office hours? I would love to. I would love to have Dixon back for office hours. I have not had a response since he was on TWIV. I don't know. It was a couple of weeks ago, right? I text him, I email him. He doesn't answer me. I haven't heard any bad news, so I assume he's just busy. I think it's time for him to be back. It's been like 6 months. So let me try and get him back for next week. We will do that. And then, Shan, you could ask him the question directly if I haven't already. If I haven't already learned. Yep. All right, one last home remedy for hiccups. If you breathe into a plastic bag tightly as oxygen converts to carbon monoxide, your body becomes more interested in oxygen. No joke, hasn't ended my hiccups many times. Well, I'll try a paper bag. Do you have a hiatus hernia? No, I do not. I absolutely do not. Yep. I wondered about that because I had an uncle who had a hiatus hernia, and he used to hiccup all the time. But no, I don't. I know that for sure. As long as I can get in touch with Dixon. Yeah, I mean, he hasn't answered my emails or text, but sometimes, rarely he does. So I will make an effort to get him on. Don't worry about it. And then he could do all the if I happen to have hiccups again, he can do all the talking. Okay. Will says, huge thank you for clarifying the difference in protective effect of vaccines against severe disease and infection. Makes it crystal clear for us lay people. Blessings. Thank you, Will. I'm glad it was good. You know, I try and keep it simple, but I learn as each try I make, I learn. And that's important, right? To hear what people say, like Will say, I don't know what you're talking about. Or what don't you understand? And if you tell me, I'll try and fix it, because I'm an educator at heart, right? I realized that with the genesis of Microbe TV. That's what I like to do. And when I did research, I taught people also, right? Taught them how to do research and communicate and so forth. So it's an extension of my research career. But I love being clear. And so I don't take offense if you say, I don't get it. You didn't explain it well. Just be nice when you do it, okay? Because if you are insulting, then I'm kind of going to put up a wall, right? I guess. Swallow ice for hiccup. My ice is big. Those crescents in the damn freezers, I hate them. I can't swallow. Maybe if I cut it, it'll be sharp and I'd be afraid to cut something. No, I don't think so. Patricia, my grandmother taught me the teaspoon of sugar trick. I've never seen it not work in over 40 years. So this got morphed into a hiccup session. I'm very sorry, folks. If it's so annoying, I won't do it again until they stop. Here's a good question from, you like to hear about Dixon's fishing tips? Sure. We'll do that. Will says, I'm confused by the effect of interferon in infection. Sometimes papers show it's beneficial, sometimes not. Okay, here's the story, Will. So the best effect of interferon comes when a virus infects you. Within hours, the cells detect it. They make a lot of interferon, and that protects the infection. That prevents the infection from going any further. All right, and that's it. That's beneficial interferon. It's on and off, because interferon is not supposed to stay on for a long time. But there are some people where the initial interferon response is too low. It's ineffective at blocking infection. So infection continues longer and to higher levels, and that keeps stimulating the interferon response. And then you have inflammation, which causes disease in itself. So if you have an initial high virus titer replication because the interferon response was poor, the interferon response will keep kicking in and not be regulated, and that causes inflammation, which causes the problem. So that's the difference. Now, why would you have a poor interferon response initially? One reason is that many people, in 20% of the population, make antibodies to their interferon, so the levels are low, and it doesn't do all the work it can to turn off virus replication. So virus keeps replicating more and more. It stimulates the production of interferon more and more, and you get into that cycle. So that's what's going on with interferon, sometimes being good and sometimes being bad. It looks like we have no more questions. Think of the last time you saw a horse with white ears. Is that going to focus me so I'll forget about things? I'm glad you've all tried to help me with with hiccups. You know, what was I going to say? I want to find a home remedy that works, but I know it's kind of contrary to what I do as a scientist, right? With with controlled experiments and controlled clinical trials. But doing the lecture helped a lot. So focus really is it. Shen says it's easily tolerated. I'm an Aussie. You mean hiccuping? Aussies hiccup a lot. That's cool. My dad, EHS says, my dad always said, as soon as you guess who is thinking of you, the hiccups will stop. Well, that's all of you, right? Because you're watching me. So, did you watch the 2023 Nobel Prize in Physiology or Medicine? No, I didn't. Should I? Should I do that? I'm happy to do it. I love that. Patricia wrote a letter to Dr. Griffin. It wasn't selected. I don't, I select the trade secret here. I select all the letters to save Daniel time. So, Patricia, just resend it. Tomorrow's Thursday. We're recording. If you send it, by tomorrow afternoon, let's say by noon Eastern, I'll pick it. Okay. But one of the criteria is length. If it's really long, it's hard to read. And so if you could make it a little shorter, that would be good. But I will read it if you send it tomorrow. Okay. Promise. Oh, okay. Thank you. Margot. Yes. You had an earlier question. He says that mandating vaccines is reducing vaccination. It is making more anti-vaccine sentiment. This is exactly what Paul Offit has posited on Beyond the Noise, that mandating COVID vaccines led to more anti-vaccine sentiment. And there's some evidence for that. So, but it was a problem. We were in the middle of a pandemic with a brand new virus that no one had immunity to. We had no real good sense of how lethal it was. We just wanted to protect people. We made some mistakes. Many countries did not mandate vaccines, yet people decided to take them. I don't know what would have happened in the U.S. if let's say a business, with hundreds of employees going in one building, said you could not be vaccinated. How do you think the people who were vaccinated will feel about that? Well, I guess they would be protected. I kind of agree with this idea that the mandates were maybe counterproductive. And you can see the pushback against Tony Fauci as blaming him for that. And he should go to jail in all of this for taking away liberties. I think we could have not had mandates if we had better communication on how to use the vaccines properly to prevent disease, and saying they don't prevent infection, folks. You're going to get mild disease, but you're going to stay out of the hospital. And it's a good thing to do. Maybe that would have been better because now we have a problem with other vaccines being rejected. Measles, mumps, rubella vaccine more and more, and having more and more measles in the U.S. So that's my 2 cents. Okay, I'll take a look at it. Scientific Library. Let's see. I'll write that down too. Drew Weissman, Nobel lecture. By the way, he will come on TWIV. I just have to schedule it. That would be fun, right? Okay. Patricia said, thanks. It is very long. We'll edit and resend. Yes, that may be why it didn't happen. What's the selection value for the 20%? I don't know, but I think that this is a product of somatic hypermutation that randomly makes antibodies to even your own proteins, and that process may differ in different people. I don't think there's a selection for it. I think it's a random process of just making the wrong antibodies. So Steph says it's not about focus, it's about distraction. Yeah. Yeah. When I get distracted by good questions, which most of them are in a lecture, you're absolutely right. Bad Cat wants to know, why are flu vaccines updated during the season? It's just a matter of how much time you need to make. Well, first of all, there's a northern and southern hemisphere, and they have winters at different times, right? So when we're having summer, they're having winter. So we look at their season and try and predict what's going to come to us. And then once we do that, it takes, 8 months to make a vaccine. If we could do it faster, which maybe we could by different modalities, maybe we could get a vaccine out earlier, right? Or make multiple vaccines as the virus changes. If we first make one that doesn't match and we say, oh, it should have been this, we could change it. But we have to have manufacturing flexibility. Fisto says, clearly a case of HPV, hiccuping professor virus. Yeah, it's very good. I like that. And Shen says, no, I mean, please do your lives with hiccups. It's human, as we as an audience should tolerate your condition. Empathy. Thank you so much. I appreciate that very much. It's very nice of you. Who are the hosts on TWIV tomorrow? I can tell you. Let's see. Go to my Google Doc TWIV schedule. So Friday, you mean? That's when we're recording. The hosts Friday are, besides myself, Alan, Brienne, Rich, and Dixon. Oh my gosh, Dixon put his name down on the schedule. He's alive and well, so I will send him a note saying thank you for being on TWIV Friday. Do you know about this ectoparasite that looks like a tank? You call it the black sphere. Yeah, he'll know it right away. Absolutely. Good. Laura says, thanks for the interferon info. It's definitely ubiquitous. Now I know why. My favorite amaretto cookies are made with apricot pits. Would they have interferon in them? No, they shouldn't have interferon in them. It's a mammalian thing. By the way, the antibodies to interferon go up as you age. The percentage of the population that have them goes up as you age. So they're the highest in the older population, which partially explains the age-related increase in COVID severity. There's anecdotal evidence that upholding an anti-vax position makes you unbearable. I don't know what you mean by that. Vanity says, did anyone see that crazy video of RFK talking about finding a bear cub on the road that he put in his car? He wanted to eat it, but his day was too long, so he dumped the body in Central Park. Creepy. I don't know if it's right. I haven't seen it. I don't know anything about it. Tom says, I think the question was about Campbell overly focusing and hyping on people's alleged physical reactions to vaccine, speaking misinformation about alleged dangers. Maybe that's more like it, because the idea that mandates are a problem is not something he would think about. But what you're saying may be the case, yeah. Raise 11, thank you for your contribution to science communication. We really appreciate it. Okay. Yes, Yu Xing, you had another request. I remember. This is another good one. We can do this. If I have Dixon next week, we won't do it next week, but maybe in 2 weeks. You want to know VSV oncolytics. So we'll talk about the development and the clinical trials. By the way, if you have any requests for a mini lecture, as Yixing is doing, make it. I'm happy to accommodate. It's another thing we can do on these office hours, as long as it's in my wheelhouse, because, I stay in my lane. I try to, which is viruses, right? So I'll be happy to do what you guys want. No, I try and notice they have left. They've left because I'm being distracted. And now it's about this RFK story. All right, so Rio, I shouldn't really bring this up because you're not being fair. You say, do you think rushing to approve 3 vaccines and then having one removed from the market with little explanation might have something to do with vaccine hesitancy? So they were done very quickly, but everything was done properly. They did a phase 1 with the right numbers, they did a phase 2, and they did a phase 3 with the right numbers. They just did them in a way that never been done before, because otherwise a lot of people will die. And they all looked great, but then the numbers are low. They're 40,000, which is a great phase 3, but it's not enough to pick up a side effect like myocarditis, which is 1 in 100,000. There's nothing you can do about that. You want a vaccine, that's what you have to put up with. But remember, the risk of myocarditis in the mRNA vaccines is much lower than the risk of myocarditis after COVID. Now, the one that was removed was the adenovirus vectored vaccine that caused blood clots, some of which were lethal. And again, in hindsight, we look and say, yeah, we might have been able to pick that up, but not in people. You might have been able to see it on the virus, and there's no way you can prevent it. And that's the topic of a recent Paul Offit column. Can we improve on vaccine? Can we reverse vaccine hesitancy? I think we didn't communicate well about them. I agree that was part of the problem. We should have said, these are rare side effects that we're seeing, and there are rarer ones that we might not see. And we think that they outweigh the risk of getting real COVID, but you have to get enough good information to make that decision. And I don't think everyone did. We tried to on Twiv, but not many people listen, as you know. But I don't think they were rushed, because nothing wrong was done. They did everything that was done in a longer clinical trial. They just compressed the timeline. But I do think that the adverse events and the mandating of vaccines were a problem. Yes, I agree with that. Oh, now we have a lot of questions pouring in. People get turned on in the last minutes. Okay, so let's read them. Oh, you wanted to do one on MERS. Okay, we could do a MERS. MERS also. Yeah, that's an interesting one. I can put that together easily. Peter S. says lots of people generalize the rare reactions to the J&J vaccine as something you could expect with all vaccines. We needed lots more public education on major media. The disinformation got ahead. Totally agreed. Major media cannot make more public education. They don't know how to do it. They need to partner with Microbe TV and other similar organizations. I don't know what other ones there are, for viruses anyway. They need to partner with us and develop something together, because we have the knowledge, but they don't want to do that, and I don't understand why. Thank you for soldiering on. You like the mini lecture. HPV vaccines. Well, why are they so good? Yeah, we could do that. 100 can potentially 100% prevent cancer. So we have a TWIV coming up, which I recorded in the Karolinska in Stockholm, where one of the guests talks about they have a program to eradicate uh, cervical cancer in 5 years because it's all caused by HPV. Very interesting episode. You'll like it. Bad Cat says, by the time vaccines available, authorities lost credibility from arbitrary lockdowns. Also, initial rollout was political, not for the dying elderly, and also close mass mandates, obviously silly. In retrospect, it's all true. The lockdowns, there wasn't a lot of time to figure out what to do. They had to react without a lot of knowledge. And remember, a lot of this was in the pandemic plan that was discarded by the former president at the time. So, yep. Spamlet, I think you did a TWIP case history of someone who got a brainworm from preparing their own bear burgers. That's right. It was a pretty recent one, right? Vincent, continued from last week. You said for a person fully opening up, not to look for SARS-CoV-2 antibodies. Can I ask why? Just thought it better to catch early when antibodies haven't waned. I'm just not convinced that antibodies are the only component protecting you against severe disease. You may have low antibody titers, but still protected against severe disease by, for example, antibodies that don't neutralize, but they still bind the virus and, say, have it taken up into a macrophage and destroyed, or from T cells. There's a lot of good evidence now saying that T cells can play a role in even protection against severe disease. So I don't think the antibody titer is the whole story. But let's say it doesn't hurt. You could do it, but it's not going to hurt, but I don't think it's informative for those reasons. Yes. Did you hear Olympic swimmers suggesting not washing their hands to try and get a dose of microbes before swimming? Yeah, one of the swimmers said he does not wash his hands after going to the bathroom to get himself a little E. coli inoculation. Well, you're going to get yourself a urinary tract infection also, for example, if you don't wash your hands. So I don't think it's a good idea. And E. coli isn't the only bacterium in the sen. So that's silly. I'm able to send the photos I have of this. Yeah, send them to twip@microbe.tv. Thank you. Yeah, eventually everyone's going to get SARS-CoV-2. But I think if you are immunized anywhere in the first 3 months, you're going to have good protection against even moderate disease. And then after that, severe disease, as long as you're not over 70 or if you don't have any comorbidities. And if you're young and healthy, I think even after 3 months beyond vaccination, you're going to be protected against severe disease. Eric says a professor in Oxford reported questions about COVID mid-December. When did the China Communist Party know about COVID? Well, there were unusual cases of pneumonia, but there wasn't confirmation until towards the end of December. So I think the Communist Party was out of the loop for a while. So I think mid-December is not right, but I'd have to check. Spamlet says showing people proof that their favorite doctor with millions of YouTube subscribers is misinforming them just makes them even more trusting of the charlatans. That's true. They don't believe me if I don't even talk out about most people. I mean, the rebuttal we did of the Chan Times opinion on the origin. We had to speak out about that because that was just 105% wrong. But yeah, if I speak out against any YouTube doctors, first of all of you here today already know it, and none of his believers are going to listen to me. So it's really a waste of time. And yes, Dan Wilson does a lot of really good videos about the trials. He does a lot of debunking, and so that's another person you should listen to. And but again, not a lot of people do. I mean, his numbers are growing, but they're still less than ours. We just passed 129,000 subscribers on YouTube, folks. Yay! And 1,000 more. We had 130,000, and then not much more to 1 million, which is my goal. Andrew Ramboat on Twitter. Yeah, I would love to get him. He's a kind of a shy guy. That would be Tweevo. That would be good because Nels would be the right one. Have you done any shows on magnetosomes? Yeah, we did a bunch on TWIP, not a bunch, at least one. You could search and find it. Yeah. And finally, Lee says, I know I'm a few minutes over. I'll wrap it up. But, we got second wind in the questions here. I don't know why. Maybe because I stopped making noise. I'm still surprised they have not gotten COVID unless it was symptomless. I've been around of any people with COVID too. I probably just jinxed myself. Well, there's some people who never seem to have caught it, and then in the last weeks they have because a lot of people are infected. You may have had an asymptomatic infection, or you may have resistance for a variety of reasons. Not inconceivable. And finally, our last comment for tonight. It's easier to fool people than to convince them that they have been fooled. That's Mark Twain. And that will do it for another office hours. Folks, we had moderators here that I want to thank very much. Vanity Nutrition, Steph SF, Les, Tom. I believe that's what we had tonight. Thank you all for moderating, and thanks to the 150 or so of you who came tonight, asked great questions, tolerated me. We have 151 viewers and 151 likes. Thanks so much, folks, for doing that. I really appreciate it. Our next live stream will be next Wednesday. That is August 14th, 8 p.m. Eastern. I will try and get Dixon Despommier here. No promises, but he's usually okay in the night. I will ask him tomorrow, and hopefully no hiccups. They seem to be gone by now because you made me very happy. Once again, thank you very much. Thanks for coming, folks. See you next week. Until then, please be safe. Good night.